All right. Welcome to the afternoon. Thank you. And we're going to wrap up the last of our team presentations. This one on the all about the prebiotic chemistry perspective on the abiotic background with Maren Frankel-Pinter and Jim Cleaves. So it looks like Jim's starting out.
Okay. Oh, well, good afternoon, everybody. Welcome back from lunch. It's really a pleasure to be at a meeting like this right now with all the chaos that's going on in the world, to be able to be among you all and share in these intellectual discussions. It's just such a pleasure. So I really want to thank the organizers for putting this together and for having me along. So I don't have a deep background in icy moons. I do have a deep background in mucking around with complex organic mixtures and trying to analyze them. But I was asked to talk about, together with Maren Frankel-Pinter, who I'm sharing this talk with, and I'm delighted to have the privilege of doing that, talk about some perspectives of the origin of life as it pertains to this question of whether there is an independent origin of life on icy moons. I will also say that that sodium hypobromite joke is going to be my big takeaway from this meeting, so thank you. Okay, so I've been working around the field of origins of life for 29 years, and the little voice in my head told me that's not possible because I'm only 25. And I wish I could say that I knew more about it than I do, but I'll tell you some perspectives that I have on it. So I think one of the things we really think about in studying this is on Earth. We've been mainly considering, how did life on Earth start, because we're here. We think about a lot of this science in terms of early Earth systems and early Earth processes. And when we start thinking about icy worlds, we have to think about kind of different geochemical settings, some of which there are cognates for on Earth and some of which there might not be, right? So things like high pressure ice interfaces and so on. Different kinds of ice dynamics that might be kind of hard to study in the laboratory and so on. But they're certainly different, and there's a diversity of geochemical environments we need to consider. So I'll just give you an overview of some things we're going to talk about. First of all, we always have to get at this question, even though it's kind of tired. What is life, right? What is it that you think you're looking for when you go somewhere? What do you expect it to be like for the sake of being an analytical chemist and trying to detect it? There have been a whole variety of hypotheses about the origin of life that have been bandied about over the years. Things that heterotrophic hypothesis versus autotrophic hypotheses. The very idea that you must have liquid water in order to have life, you must have organics, you must have some source of nitrogen, and so on. I will say, when I entered this field, there was kind of a burgeoning, what would you call it, feud between people who favored these sort of hydrothermal vent origin of life models versus the Miller-Urey type people. And it's really nice for me to hear in this community that it's like, well, what if we take a comet goo and put it in a hydrothermal vent? And it's like, yeah, well, that's the best of both worlds, right? So I'm glad to see there's some progress in that field. So there's questions of compound inventories, what we heard in the last pair of talks. There's a great diversity of different types of organics we might want to use as our toolbox for assembling life. There are commonalities in some of these processes and very significant differences in others. There's this dependence on planetary, or I'll call it moonitary history, right? The different bodies will geochemically evolve in different ways. I'm hesitant to use the word lunar. It sounds too Earth-Moon centric. So what is the baseline range of abiotic chemistries? Do we have that really mapped out well enough that when we see something anomalous, we'll be like, well, that couldn't possibly be explained by a geochemical process, right? And then we have these kind of questions of culling and complexification that lead towards the organization of matter to a state we might call living. And I think Maren is going to particularly talk about a lot of these concepts. And so where we really want to end up is, how do these ideas of the origin of life feed into the concept of life detection, right? So this question of what is life, I don't know. There literally are books that have been written where they just collated the essays of hundreds of scientists and called it "What is Life?" and you read them and see what you think. There's actually a lot of overlap sometimes in definitions and what people think is the sine qua non of life. We have Virchow's concept of all cells come from other cells. So this is even before we really had a deep understanding of the molecular interior of the cell. There was already a biological model that cellularity is the start of the phenomenon of life. We have this NASA working definition, which is a little more Catholic. This idea that it's a self-sustaining chemical system capable of Darwinian evolution. That leaves a lot of room to cram different models and molecules and environments into a broadly classifiable phenomenon. And then we have life as we do know it, which is what a lot of our analytical techniques are built around, right? Being able to detect things like amino acids and fatty acids, doing PCR, things like this. We're very good at that. But maybe we need a kind of more broadly open-ended definition to be able to look for life on other worlds. We really don't know how stochastic the process is, or if it has some sort of strange attractor nature that all life ends up being more or less like ours, right? So just a few other definitions just to point out the fact that you start seeing reiterations of the same concepts over and over. Life is self-reproduction with variation, shorthand for Darwinian evolution, potentially self-perpetuating system of linked organic reactions catalyzed stepwise by specific organic catalysts produced by the system itself. Yeah, that sounds about right. We see these kind of things. It's very much trying to pigeonhole a lot of terrestrial phenomena into a very inclusive kind of concept, right? Okay, so I do like during the 60s and 70s, we had the emergence of this concept of system theory, general system theory from von Bertalanffy and so on. And this idea that a system is defined as a regularly interacting, interdependent group of items forming a unified whole. The system is described by its borders, by what crosses the borders, and by what goes on inside. So the point of this kind of things is that can describe a Boy Scout troop as much as it can a cell, right? It can describe many other phenomena, and you can talk about the dynamics in a very abstract way. And I think around that same time, we had the chemoton theory of Tibor Ganti in Hungary, which broke life down into this kind of three interconnected processes, one which does work, one which encapsulates the system, and one which stores information about the system. And the three of those subsystems are interlocked, right? So what that's saying is you can't, once you take any one of those parts away, it is no longer a living thing, right? It's a system. So I think that's important to think about, in terms of looking for molecules, that maybe it's not a molecule that we're looking for, it's a system of molecules, and to keep that in our minds. I was asked to kind of just dump into this definitional question. We often bandy about these words biotic, abiotic, and prebiotic. Biotic, we say, is things that are derivable only from biology, that we know that. Abiotic would be compounds or processes and so on. Things that are unequivocally not attributable to biological processes. And prebiotic would be those things that we know are precursors to biological systems, right? Since we don't know how life started, you can debate whether the concept of prebiotic chemistry has any meaning whatsoever, right? And people interchangeably use them, and I'm not the grammar police, so do whatever you want to do. But I think it's important to keep that in mind, that when somebody says something is prebiotic, okay. Okay, so what do we know? Is the origin of life fast, easy, hard, slow, what? We have the general belief from molecular phylogenetics that all life on Earth is descended from a LUCA organism, a last universal common ancestor. That last universal common ancestor was very much like a modern prokaryotic cell. putting together molecular clock evidence and the geological evidence, we think life started somewhere around 3.8 to 3.5 billion years ago, which gives you somewhere between 500 million and 1 billion years for life to start on Earth, right? Somewhere in there. But we don't know if it took that whole time. Maybe it took 2 weeks, right? It could be some incredibly easy thing, and we just had the right set of conditions on Earth. Bingo, there it goes, right? Important to keep in mind because it turns out now you can do informatics on chemical databases like SciFinder and Reaxis and so on. That 98% of all the chemical experiments that have ever been reported in the literature are done for 48 hours or less, right? And that's, it makes sense. What you do, you turn on the hot plate before you go home, and then you turn it off in the morning, right? Or you turn it on Friday and come in on Monday. So we don't really have a great deal of experience with very long, slow chemistry, at least in manipulating it. This topic came up again in the context of Bennu recently. I saw on the news. Here you have these bodies that are full of organic compounds, including ones we think are important for life. And we think they've been going through aqueous alteration for tens of thousands, maybe some places hundreds of thousands of years, but they don't seem to have life in them, right? So what the heck, right? That's time, water, and organics. And, right? So one of the speculations that came up is like, well, maybe there's a specific niche environment on Earth that doesn't exist in those kinds of bodies. And one being possibly the prevalence of sunlight, right? That photochemistry may be very important for driving the chemical disequilibrium that starts life. And we think about that. I mean, the Earth is highly insulated. The outer planets much less so, right? I don't know if you need light to get life started. I don't know how much you need, but that could be a problem. I don't know. Then again, we have this concept that there may be very Earth-like hydrothermal flow underneath the ice surfaces of these things. So there might be exactly the kinds of niche environments we needed to get life started on Earth. So that's an open question. On the other hand, it's very possible, and we may find this out, that these things are just giant tubs of cyanide polymer, but that's been sitting cold in space for billions of years, right? That's possible. Okay, so the thing we really wanted to stress is the kind of importance of the interplay of the input organics, the environments, the way those environments shape the way those organics develop, sort of primitive autocatalytic reactions and how those complexify are all happening in some sort of dynamic interplay, right? And you may need to match all of those things in a very specific way to get replicating systems robustly kickstarted. Okay, so I do want to bring this point home. Habitable environments aren't necessarily inhabited, right? The origin of life may require something that doesn't exist on a planet that's otherwise perfectly habitable, right? If you have good sterile technique, you can leave your Petri dishes sitting at room temperature for years and years and years. If you have bad sterile technique, well, there's opportunists, right? And whatever conditions were required for the origin of life on Earth might not be the general conditions required for alternative origins of life, right? We don't know. I do like this point. Somebody brought this up a long time ago. This is one of Louis Pasteur's swan-neck flask experiments, which were crucial for settling the abiogenesis debate in the 19th century. These are open to the air. It's sterilized hay infusions, right? This has been sitting open in a glass case in Paris for 160 something years, right? It still looks pretty clear to me, right? So that's full of biological precursors, right? It's full of fatty acids and amino acids and probably purines and pyrimidines. It hasn't self-assembled itself back into life yet, right? So there's a data point. I don't know what it's telling us, but it's telling us something. Okay. So general model for the origins of life, have some starting environmental process that creates chemical disequilibrium, introduces energy, that makes some kind of organic precursors. Which ones of those you think are important now becomes an open question. Those react to make some kind of dynamic building block that allows you to construct a more complex kind of adaptive chemical system. Again, which ones of those you think are important becomes an open question. You have some processes of culling or natural selection that make some of those fitter than others, and they become endowed with the ability to self-reproduce and bootstrap themselves from their neighbors. And then you really have something, this little blue arrow right here is what I have spent 29 years of my life trying to understand, is how do you get coherent, naturally selectable chemical systems from this kind of random chemistry, right? This is really the crux of the problem, in my opinion. And then if you go on through some time, you get to very coherent, naturally selectable systems, right? Like things that have very robust genetic codes and error correction systems and so on. Things that we might not expect to fall right out of the closet at the first go, right? And this is really neat. This is actually, they just discovered this last year. This is the oldest known example of the precursor of all alphabets on Earth. It was found outside Aleppo, Syria, and which is really awesome too, that the first thing people were worrying about was chemical analysis, right? 4,500 years ago. CHON, right? I guess it's Proto-Aramaic anyway. But so, you know, from these initial kind of scrawlings for storing information, we don't even know what they were scrawling. It wasn't full grammatical sentences. Maybe it was keeping track of debts or something. You know, this evolved into a very codified languages with very exact grammars that are recorded so posterity can really understand them. The point is that the very primitive version of the thing may be hard to recognize, right? Unless you squint or open your mind a little bit. So, and you know, the other important part of this from an organic geochemist perspective is that if we have this perspective that the point of prebiotic chemistry is to make biological precursors, the fact that biological things release those into the environment when they die means that searches for those things are going to converge on the same positive signals, right? And we need to have a way to differentiate, which is the point of this workshop, what is contamination and what is background abiotic chemistry and what is really some sort of internally directed disequilibrium. So in the context of icy moons, I like to bring up the Arrhenius equation. Obviously, we've seen from yesterday several talks that we could possibly expect very warm water at the bottoms of some of these oceans, but things are just generally slower the colder things are, right? So we might expect chemistry in general to be moving at a slightly different pace than it would on the early Earth, right? Some people believe that the early oceans were around 70 Celsius. If that's the case, you know, just the kinetic differences in chemical evolution might be 100-fold different. And in that sense, you know, time is money, so to speak, that things are just moving faster because the environment is more kinetic. And I will point out, this is the Dobert finding that you can actually measure the temperature by the rate of chirping of crickets, all right? And this was reported, I think, 1897 or something. And it was reported in Fahrenheit, just to, like that. I don't know, it still works, right? Okay, and so this is a phenomenon. I just want to point out, if we look at the diversity of terrestrial vertebrate species on Earth, they're the hot spots around the equator, right? The pace of evolution or the pace of speciation in certain clades is really related to the flux of energy through the environment, right? That's something to think about if you're going to a low energy environment. But how is it working? Okay, so there we go. With the sample size of 1, we have these concepts of cells, the importance of genetic information, the importance of metabolism, and the way we can understand that in the light of terrestrial biology. Our concept excludes things like viruses, and prions, and viroids, and so on. Are there other concepts that we haven't even thought about because they just don't exist here? I don't know. Again, I wonder how much the legacy of Virchow and Schleiden and Schwann and so forth have influenced our thinking about what life can be like. We do think it has to be cellular for chemiosmosis and all these sorts of things. Maybe there's space for more imaginative thinking. Okay, so basic concepts that we think are probably important. Cellularity, I'm going to say that again. I still think it's real, and I think it probably has to be at a certain scale that's governed by physics and chemistry. Catalysis is almost how living things turn themselves into time machines. Heredity does seem logically like you have to have something like that. But there are probably a whole bunch of different chemical ways of engendering those phenomena, right? We already know from chemical studies that there's 20 some odd perfectly valid ways of making genetic molecules, many that can be copied, many that can interfere with biological information expression. You know, we're limited by the number of people exploring chemistry in that sense. So what are the elements of chemical evolution? What seems to be important in many types of evolving system is that you have selection that can occur on multiple levels. So you have hierarchical nested systems, some ability to store combinatorial polymer-like molecules that can be either catalysts or information storage. This concept of open-ended or near open-ended exploration of possibility space, which I think, given organic chemistry and some of the talks we saw earlier today, that goes hand in hand with organic chemistry. So not something we necessarily have to worry a lot about. But the possibility that a dynamic, challenging environment is really one of the principal drivers of not only biogenesis, but subsequent evolution is an important thing to think about. And these other aspects of storing information and doing work, I think, are, you can pick them apart. It's hard to imagine a non-machine-like biology in my mind. So with that, I'll just end some work we did a few years ago on trying to see if we could use machine learning to pick out, to distinguish between biological and non-biological complex organic mixtures just based on their mass spectra, right? And so we used high-resolution Fourier transform ion cyclotron resonance mass spectrometry. And you can see these are spectra. It's Exxon Petroleum, I think it's a well off the coast of Venezuela, E. coli cell extract, Murchison meteorite extract, and a Formos reaction mixed with ammonia. You look at them, they're all pretty complex spectra, right? There's thousands of peaks. I don't think you could just look at that and be like, oh, that's biology, that's oil, that's that. Maybe if you're a very trained organic geochemist, you could, but there's plenty of data for a machine learning algorithm to find signals that differentiate things. And these are Van Krevelen diagrams, transfers of this data. And you can start to see trends in the way the organics mature, general loss of volatile elements and so on. But what is significant here is you can pick out the isobaric species for different types of biomarkers. So these are diketopapyrozines, which are the breakdown products of proteins. So we can find the individual masses of all the possible, I think it's 400 dipeptide diketopapyrozine isomers in that neighborhood, or straight chain fatty acids, which are parts of cell membranes. And you can see we can even pick out the contamination in these samples. So this is a fingerprint, C16 and C18 fatty acids that just get into everything, right? You touch a test tube, you've transferred some of your finger grease to everything. You get samples somebody collected in the field. People don't wear gloves. Even if they did wear gloves, there's anti-slip agents that are put on gloves so that they come out of the box nicely, and that is oil. The classifier still can look past that contamination and find the signal in these complex things. So I think that's all I want to say, and I'm going to pass it off to Moran. And we'll take questions at the end, I suppose. All right. Good luck.
Can everyone hear me well? So I'm Moran Frankel-Pinter from the Institute of Chemistry at the Hebrew University of Jerusalem. I'm really happy to be here, and thank you again. I also want to thank the organizers for this, and Svet and the organizers for this amazing workshop, and I really learned a lot. So following up on Jim, we know by now that the building blocks of all of today's biopolymers, so of proteins, of nucleic acids, polysaccharides, the most abundant biopolymer, they're all formed we see them in model prebiotic reactions, we find them in meteorites, and now also on asteroids. But in addition to finding, I guess, the building blocks, we also find a huge variety of other important molecules that did not make it to the biopolymers, but they're part of metabolism, or they're just not in biology as we know it. And there are many questions about, how do we get from simple building blocks into these polymers via various pathways? And one question was also, so lipids are not polymers, but they're really important biomolecules today. A big question was, for example, phospholipids. They're probably late, but now, with Ram's work and other works, we now think, well, maybe not so. Maybe they're not going to be, these biomarkers. Maybe it's easy to make them abiotically. And we've heard, of course, about abiotic, or I guess prebiotic or abiotic, how we want to describe it, chemistry, generating a lot of the molecules that we know. And we know that the prebiotic chemistry work really, that gave rise to a lot of what we know about prebiotic chemistry was from the Miller-Urey experiment. And as was discussed, we know that just really taking a very simple mixture of gases with water generates, in a very simple apparatus, amino acids. But we now know, so back then in the 1950s, they used really simple technique of 2D thin layer chromatography, and Miller described 5 amino acids that they've seen. Now we know, of course, with analysis, there are so many molecules in these experiments and other apparatus that has been used. But I think that one of the maybe consequences of this chemistry that was back then, in the 1950s described, is that maybe it led to the parsimonious idea that we still have, that, okay, it's really easy. We get the molecules of life, so it's just going to be super easy to get to biopolymers and also to life. But now we know, of course, that it's not that easy, and the path may be very, it's going to get, in order to get to life, which we don't know what it is, maybe we need to look at many alternative or orthogonal pathways. And this was also described when we look at the abiotic chemistry or prebiotic chemistry or asteroids, meteorites. We find a lot of the amino acids that we have in biology today. We said that we have 20, of course, proteinogenic amino acids that we have in today's biology as we know it. But we find, usually we say 10, but we know we've heard from Heather and from Gregoire and Alexis, we've heard about there are 14, for example, amino acids that are proteinogenic in Bennu, right? And many other ones. So hundreds and potentially thousands of amino acids. So one of the big questions is that when we think of understanding the path from simple molecules to more and more evolved species, then what would be the selection mechanisms, and Jim talked about this too, what would be the selection mechanisms that lead from this pool of different building blocks to very specific patterns or enrichment of some building blocks over others? We've also talked about sugars. Now, this is kind of interesting to me that, so yes, we know that we've had many, sugars. We see them in meteorites and so on. And Ram tells me that we need to modify, soon we'll see the paper, that we need to modify what we think we know about the Formo's reaction. But so we know, yes, we have sugars. Now, most of the work that people have investigated in the context of sugars in abiotic chemistry is going to RNA, why ribose and not other sugars? How do we get from, so we have the building blocks of nucleic acids, or we have sugars, we have phosphate, we have nucleobases. How do we get to, for example, RNA? But almost no studies have been done on how do we get to polysaccharides. This is the most abundant biopolymer in today's biology. So this also brings me to the point of, are we directing, the research here, for example, towards certain molecules? Yes, but there are many other directions that even biology today tells us that might be important to look at and interesting. Also in the context of RNA, we also talked about, purines and pyrimidines that we find. But in addition to the pool that we see today or we see in meteorites, there have been so many other options, right? So many potential, even if we think of a nucleic acid kind of molecule or a replicative molecule. Yeah, we know the chemical space was huge. So why should we focus on a certain set of nucleobases, on a certain set of sugars? We can explore the huge space. Why do we think of a nucleic acid backbone? There are many other ones. So yes, people are addressing this, but I think not enough. We also know, so this work from Ram showing that if we have chimeras, for example, of nucleic acids, then we're going to see basically building up of homogeneous chimeras of nucleic acids. So interesting question is also how can we get from a more messy mixture to a more homogeneous or mixture through different pathways of chemical selection? Questions about the evolution. This is an example of evolution of peptide backbones that people, myself and others, have utilized to look at evolution of peptide-like molecules. So in this case, we have mixtures of amino acids, but also hydroxy acids, these similar analogs that contain alcohol instead of an amine group. And as we do dry down reactions, we see formation of deoxypeptides or molecules that look like peptides. But what's important here is that the evolutionary pathway, so if we accept that there are many other polymers that can be formed, we can ask the question of what would happen as these molecules evolve. This would be due to, what would be thermodynamically more stable or kinetically accessible. And as we do these experiments, we see evolution of the backbone towards, in this case, to what we looks more like peptides. So enrichment in peptide bonds over other bonds. And this I brought up, so as we think of compartments or how to make, yes, the amphifiles. So we talked about phospholipids, and maybe they're not so late. Maybe actually it's really easy to make them. But we know that assembly is cheap. So even if we just have very simple amphiphals, that amphiphilic molecules that we have in meteorites, like fatty acids, they would form a variety of structures, micelles, vesicles. We can alter their morphology. And this shows the abundance of 2 to 10 carbon length fatty acids in different meteorites. This is just to show that, yes, we find a lot of these simple amphiphilic molecules in meteorites that can also, the long ones, can also form very robust structures. But, and this was brought up yesterday, if we think of amphiphilic molecules as biomarkers, what should we be looking for? So yeah, in the abiotic background, we see these one carbon, I guess, spacing. So we see all of these species with one carbon addition. But we know that in biology, due to biosynthetic pathways, we have an even number of spacing for the long amphiphils. So this was brought up, yes. So should we be looking for these signatures that are, not one spacing, but a double spacing, something that looks different? Also, something that was brought up yesterday was maybe looking at, interesting branched, lipids, or we know that in biology we have generation of some double bonds or cyclic lipids. So maybe, all of these kind of signatures, yeah, that we would not expect abiotically. Okay, so in general, when looking at the question of how can we look at the transition or study the transition from prebiotic chemistry to biology, as Jim mentioned, we have so many question marks, and there are so many things that we don't know, of course. We're trying to open up the black box of what happens if we take the starting point, the huge set of small molecules that we know that we likely had on Earth, and then building up complexity from there to look at how small molecules increase in chemical complexity to form functional polymers. Can we see formation of assemblies? Can we see emergence of function, which many times would be related to assembly? What is not shown here, and it's also all the time occurs, of course, is evolution of metabolism. So yes, we also had polymers and metabolism had to co-evolve, so we kept having all of these mutual interactions all the time. When looking at these processes of increasing complexity, typically what we do is we either start from here, so we can take a top-down approach or a bottom-up. So we can say, okay, from a top-down approach, we know how life looks like today. So let's try to refer back and think, okay, how can we build up the complexity, or how can we get from looking at today's biochemistry, what can we say about how life arose? Of course, from the bottom-up approach, we typically either say agnostically, we don't know, let's try. We know what we started with, so let's try to build up complexity and see which protopolymers we get, what is the evolutionary process that takes us from one step to the next, and so on. Another more direct approach is, I would say directed approach is people would say, okay, we know we want to get to RNA. So how do we do this? We don't know yet. Maybe people will disagree with me. I think we don't have a good way of, we don't know how to make RNA yet. I think it was very late. Okay. So one step is, yes, we have the huge pool of building blocks, but what can they do? And we know that important for biology as we know it is the formation of polymers. So there's a lot of research about how can we get to polymers from simple building blocks. And if we talk about, of course, ocean worlds, then we're talking about mostly chemistry that will happen in water, and there's a lot that we know about this. In the context of Earth, there's a lot of work on drying reactions, and I'll also touch upon that. But so if we think of, from simple molecules to complex polymers, a lot of the work has been done, to make RNA polymers, to make peptides. There are many mechanisms. So now, yes, we can take amino acids or simple, similar analogs combined with amino acids, and we can get peptides, or we can take the precursors. This is from Palmer. We can take the precursors of amino acids, aminonitriles, and get the formation of peptides in water. One of the biggest questions, though, that we don't know yet how to do is, okay, we get peptides, but these are short proteins. How do we get really long ones? Yes, there is always an equilibrium in water with degradation. So yes, we can make, but we still don't know how to pass this threshold from small oligomers to really long ones. Okay, so we still have this to think about. So we form polymers. Great, polymers can do things that monomers cannot, but are we going to see an increasing complexity of the system? So one thing would be to look at emergent assembly, starting with a pool of simple building blocks. Many times we don't see anything by microscopy, but once we form oligomers or polymers, then we start seeing things. This is, for example, work by Jim Cleaves and Tony Gia, showing that if we just take hydroxy acids, we dry them down, we form polymers that are called polyesters, put them in solution, and we form a bunch of droplets, okay, that coalesce and then split. So there's a lot going on there. We see this with many systems. This is from Jason Greenwald and Ronald Drick, showing that if we just take amino acids, simple amino acids, we put them with the volcanic gas carbonyl sulfide, and we see condensation, dehydration of amino acids to these short peptides. And then once we have these peptides, we see these fibrils that look very much like amyloid fibrils that we see in neurodegenerative diseases or as functional amyloids in bacteria and other species. Very, very stable assemblies of peptides. So assembly is easy, and of course, I didn't mention here, but amphiphilic molecules, even those that we just find in meteorites, form a variety of species. Now, why is this assembly important? Of course, so assembly, for one thing, assembly is going to enable us, for example, an environment that is different, that is confined from the outer environment or the bulk media, where we can potentially catalyze reactions. Another very, very important thing that we know about assembly is that it hinders hydrolysis rates. So we'll talk about this, but if we think of water, as we now know that water is not just a solvent, but it's a very important, we'll talk about this chemical participant in all of biochemistry, then water is the best friend, but is also the worst enemy because it degrades all of our biomolecules by hydrolysis. But so if we have this assembly in water, then we know that the degradation or hydrolysis rate, degradation by the addition of water molecules, is hindered if we have assemblies. So assembled species degrade more slowly compared to non-assembled species. This is a very important mechanism if we think of evolution of molecules. Assembly would be important, should be important in water for these types of reactions. I don't show it here, but there are also many examples that once we have these assemblies, then we can do catalysis. Okay, so many assemblies are catalytic. Okay, chirality. Wow, that's a big thing, right? We talked about this for so much in this workshop. So what do we think about chirality when we think of chirality from an origins of life perspective? And of course, we know that all of our biopolymers are homochiral. We know why this is. We know it has to be, it has to be at the very large polymer level. If we just consider short oligomers, then there we see a bunch of functional small heterochiral polymers. But if we think of long oligomers, long polymers, then this is where homochirality must be there in order to have function. Also, it's not possible that, I don't think, that in one planet we'll have the two mirror image. It's just not sufficient to, it will be just one, right, eventually. So first, if we have a racemic mixture, which is what we always get from chemistry experiments, if we have a racemic mixture, then first we need to somehow break the symmetry. And then there's a separate question of if we broke this symmetry, what would be the mechanisms that will propagate this asymmetric enrichment to form these homochiral polymers by replication. And for symmetry breaking, this is one of the questions, right, about meteorites and so on, and did we have an enantiomeric enrichment, and if we did have, is it stochastic, due to stochastic events, or is it due to, for example, interactions with minerals? There are now different theories about how, rock-water interactions will affect these enantiomeric enrichment, and how can we propagate this homochirality? All right. So when we think of, the rise of homochirality, and so if we did have this stochastic and enantiomeric enrichment, how can we propagate this? And from a molecular perspective of increasing complexity, I think we have a pretty good answer now. So we know that homochiral polymers are going to be more stable than heterochiral polymers. We know that homochiral polymers are going to hydrolyze more slowly than heterochiral polymers. Again, this goes back to polymers can form assemblies. Assemblies are going to be different than the monomers and are going to have a differential hydrolysis rates, differential stabilities. Yeah, if we think of, but the mechanisms for chiral amplification, so one thing that people are looking at is how can we, have autocatalytic reactions that will give us chiral amplification. And there's a very good, interesting reaction, the Soai reaction, that gives us this chiral amplification, but it is not prebiotic. So many people are looking for chiral amplification, abiotic or prebiotic reactions, in which we can see this propagation of chirality through the, products of such reactions that will amplify these processes. Okay, and again, going back, I will just say that I think, yeah, this is maybe the elephant in the room that we need to understand. So we see that in Bennu and in Rigu, we don't see enrichment of amino acids. So what is the threshold that we would expect from, due to stochastic events to see of enrichment of enantiomers? Is it 17% or is it 1%? So I think we need to understand this. And another thing I would like to talk about is really, I started talking about this, but that if we look at all of today's biopolymers, so these are the polymers that we have today, right? They're all showing us evidence for water-based chemistry. So all of today's biopolymers result from condensation dehydration reactions, so reactions that involve the removal of water molecules. If we make a bond between amino acids to form a peptide bond, it's condensation dehydration reactions. We need to lose water. Same for making nucleic acids. Same for making polysaccharides. We need to lose water. And that means that if we add water to these polymers, then we are pushing towards hydrolysis. So we need to add water in order to break them down. That means that right from a thermodynamic perspective, we are not at equilibrium at all, right? Otherwise, we would just, in an environment with water, we would just hydrolyze. So we are in a deep kinetic trap. How can we avoid this kinetic trap, going back to water as the enemy and as a really good friend? Different models have been proposed in the context of environments that do allow drying. One solution was wet-dry cycling, which would drive through us towards polymers that are going to be persistent in water and evolve through changing hydration levels. In this case, the idea is that if we have monomers that can condense and dehydrate when we dry them down, if we dry them down, we push the equilibrium towards just dehydration reactions and forming polymers. But if we then put them back in water, we're now going to have species that are going to form structures and persist in water oligomers that are not, so they are going to degrade faster than the structured ones. And if we do this again and again in this experimental platform, we are building up polymers that are surviving in water and are getting longer and longer. But how can we, so if we don't have such an environment, for example, which types of environments will drive oscillations of water if we think of water-based chemistry like in today's polymers, which is not the necessary good assumption. So of course, if we think of these other ocean worlds, then people have talked about aerosols. These would also give us environments that have low water activity. Pores in rocks, we know that in hydrothermal vents, due to the heat, we have also dehydration reactions. We heard from Jacob about these, the brine, right, the icy brines that will affect the pH, the hydration level, the ionic strength, so we can have these brine pockets where we will have dehydration reactions. Will these environments, one question, will these environments oscillate the water? Because I think, Jim, you showed this spontaneous generation kind of Louis Pasteur experiment that didn't evolve much, right? But this is kind of a static environment, so right, the oscillations in all of the environments that we've discussed, there's always oscillations of the environment, and this probably was very important to drive chemical processes. And freeze-thaw cycles, I think we don't study this enough, which could be really important for understanding what could happen in these chemical reactions. And I'll just end with this. We talked about the question, and Tori, you brought it up too, about explosion versus compression. And of course, in meteorites, we find a huge variety of molecules. I think that, but our assumption is that as we go to more and more complex molecules, we also have this explosion. But I'm not sure if this is true, because yes, we'll have many molecules, but we see in very simple chemical reactions that there is a lot of compression of the chemical space because of chemical selectivity of certain molecules over others. So even if the pool of small molecules is big, if we have limited energy, which we always have limited energy to some extent, we don't have an infinite amount of energy, then there's going to be some compression of the system. Here in this paper, we show that we took reactants that can dehydrate or link through dehydration reactions, and we added more and more reactants under lower temperatures. And we showed that as we add more and more reactants, actually we didn't get more and more products as we expected. So I think we need to question about this explosion. Is it really going to be explosion, especially if we think of lower temperatures? So we need to better model, at these temperatures, if we think of lower temperatures, we are limiting, of course, not only the kinetics, but also, as Kristen said, the thermodynamics is also going to be affected by these changes. Okay, yes, so I'll skip this, but I'll just say, how do we get, yes, to this increasing complexity without grad students, without rotovapes, without all of this equipment that we have? Maybe this didn't come off right. We need grad students. No, this is what I mean. We need to think of reactions that are going to easily get us to, that are probably occurred and are going to occur again and again, are going to be robust, okay, just with the aid of graduate students. With that, thank you for listening, and let's discuss.
We have time for a couple of questions, but we have a scheduled break now too for a few minutes before we move on to our last talk. But we can take a couple of questions. Yes.
Oh, that's
I have there too.
read it on and all.
Interesting.
Nice talk.
Well, this is online question first here, and it's from James Lewis from NASA Goddard. It says, are there studies into which amphiphile structures would be stable in the chemistries we model for the Europa and Enceladus oceans? Are there studies into which amphiphile structures would be stable in the chemistries we model for Europa and Enceladus?
Yeah, I'm gonna
Any
say
thoughts?
yes.
Yeah.
Or
yeah.
perhaps anyone else in the room too, if you.
Yeah.
Yes, you-
For example, Claudia Bonfio is doing experiment with really high pH and alterations between, let's say, pH 9 to 11, and it can cause really division of very simple amphiphils, and also the chemical reactions that she's looking at. So I think, amphiphils are just the masters of self-assembly. So just given the right conditions, or if you have eutactic conditions, then definitely you're going to form some structures, and they're going to be dynamic. Probably yes.
Given the right amphiphils too. Right?
Yeah.
Yeah.
Other questions?
This is for Jim. This is a great talk.
Yeah.
So this might be a difficult question. How relevant do you think carbonaceous chondrites are and their organic contents are to the origin of life? Because it appears that they contain almost every possible isomer, and maybe we get all excited when we find adenine or amino
Yeah.
acids because we're just focusing on detecting those.
Yeah, well, so right, you got to measure something, right?
Right.
But yeah, I thought that was a really nice thing in the previous talk, where, what was it called? You're showing that, guess the precursor, that they had these 3 mass spectra from different starting materials, and they were all like remarkably similar, right? And, I think Miller published a paper in like 1972 or something, comparing Murchison to the Miller-Urey products, and they're pretty similar, right? So, I mean, I think once you get into that kind of radical reactive chemistry space, it all starts kind of filling the same maze, you know what I mean? So I think, yeah, maybe it's very relevant because it's one example of that thing, right?
Right.
So,
Okay, thanks.
Maybe I can say something about this too. The way I imagine it is that those astro materials just brought Lego bricks onto the surface of the planet, and then whatever processes we're doing to get to this point that we call life now, then it chose among those things that were available and just optimized it. But originally, everything was there to start from, and then whatever worked went on, whatever didn't work stopped. Basically, that's the way I see things. But maybe I'm wrong.
you're saying more in the say the context of like ground up synthesis. I mean, that's the other option, right? So maybe both.
Eloy Camprubi, University of Texas, Rio Grande Valley. I just want to, since we are talking about Legos, I just want to remind everyone, we all know this, but here's the reminder of the week that building a cell is not a Lego. Polymers and molecules are very important. Molecular complexity is a requirement, but it's not sufficient. You take a pound of E. coli, you kill them softly, like the song, with like making pores in their membrane, so all of the molecules remain there. There is nothing we can do to that perfect soup to make it go back into a living system. So organics, the molecules, life is not an object, it's a process, it's a verb. So the dynamism of the origin of life is totally, I wouldn't say missing, but we need to integrate it better on the molecular complexity track. And then people like us who care about origin of life and life detection, I think it's a challenge for us to see if that can be added into the life detection methodology, which is not very easy. I know that Everett Schock has been thinking about a lot about this and many others. But yeah, anyway, we all know this, but.
I think we're gonna have to wrap it up there, so that we can finish on time today and make it to the Library of Congress. Did you have one more thing to?
It's sort of a half-formed question, so let me keep thinking.
Okay. All right, so we're gonna take a break, and then we'll have Sarah Horst. Yeah, let's thank all.